As promised, I'm doing a video podcast of the last lecture to try and catch up due to my absence. Okay, so we've already covered setting the scene in terms of potential drug targets that were available in the towards the end of the 20th century. GPCRs, enzymes, and hormone targets predominated at that time, and there's a link to talk about those drug targets and their nature. The statistics for the human genome is such that the size is 32 gigabases, which is a huge amount of information. Repeated sequences are quite prevalent. About half of the genome, human genome, is repeated. The reasons for this are slightly poorly understood. Only 28% of the human genome seems to be transcribed into RNA, and only a small percentage of that ends up as encoded proteins. And as of course we know, proteins are the major target for drug treatment. 7% of all genes are vertebrate specific, and the rest are general to most life systems. A slightly more advanced look at drug targets into the 21st century. Of the 30,000 human genes, only a minority may be interesting targets. The estimates range from 3,000 to 10,000, and as I mentioned before, that this is a wide range and quite fuzzy. This figure is always being re-estimated and revisited. Many proteins that are deemed to be of potential interest are orphan status. Sometimes their structure is known, sometimes it isn't. But their function or natural ligand, if a natural ligand interacts with those drug targets, with those proteins, may well not be known. So a lot of the effort into validating drug targets is to find their natural function, and if they interact with small molecules in the body, what small molecules they may interact with. So compared to the current state of play, at the end of the 20th century, it's at least an order of magnitude increase in number of targets. Probably an easy figure to reconcile this is this way. So if we look at 500 at the end of the 20th century, and we look at the range. If we say 5,000, that's a nice figure to probably go for. So 500 at the end of the 20th century, coming up to date now about 5,000. So it's an order of magnitude difference. Novel techniques have been developed to rapidly validate targets by trying to elucidate their physiological roles in the human body, and this involves mainly molecular biological, molecular pharmacological efforts, but medicinal chemistry approaches or chemical approaches are in there as well. Amongst these approaches, functional genomics or chemical genomics exist, and the use of chemical or bioinformatics may be used to elucidate structure and functional relationships. So actually finding what that gene product does, functional genomics, or throwing chemicals at that gene product because you suspect that they will react to small molecules, and seeing how they react is known as chemical genomics. And then that's associated with chemoinformatics and bioinformatic approaches, comparing protein structures to maybe known proteins that have known function. Part of this process is that you can compare across genomes. So there's nothing stopping you comparing human genome with mouse genome and C. elegans, and trying to achieve an idea of the function of a potential drug target, a protein, a new protein, by comparing the materials across genomes. Eukaryotic genomes are obviously more closely related to prokaryotic genomes. So comparing the genomics or comparing proteomics, comparative genomics, comparative proteomics, can throw light on function of particular human drug target proteins. Let's take a slightly different look at what's called the biofarmable genes, and this was a GSK study in 2012, and they took 13,000 potential biofarmable genes and their products and tried to classify them in terms of their exploitation and their nature. So we can see that there's a massive chunk of the pie that's called unclassified and various functions, and we will come across these types of systems as we discuss drug targets, I'm sure. Ones you can relate to more easily are enzymes as a drug target classification, the ion channels, and notice that ion channels have become more significant in the 21st century, and ion channels are being elucidated far more frequently, and people are paying much more attention to them now. And then membrane receptors, which include the GPCRs, transport systems, which move small ligands and ions across membranes, surface antigens, proteins that occur at the surface of a cell and may be involved in cell-cell recognition or cell invader recognition, things like that. Cytosolic others are in there. Transcription factors, even though that's a small chunk of the pie, it's a very important chunk. And then we have proteins that are regarded as secreted, and they're transported outside of the cell for whatever function. And then nuclear other, so things that are in the nuclear compartment within the cell, in the nucleus, and have a function specifically there. We talked about high throughput screening in drug discovery. The core format for this has been driven by robotics. It is what's called the 96-well plate. The 96-well plate is driven by the 8-bit characteristics of information recording driven by computers. And then variants of the 96-well plate include much larger formats, but they are multiples of 96, so that the data can be tracked readily. So the high throughput screening is driven by particular types of assays. So we have ion channel assays, which look at often cellular systems. So we have a cell-based assay where the ion channel is overexpressed in the cell wall, and often a cell line that's used is frog oocyte, and human genes are put into the frog oocyte, and the ion channel gene is overexpressed. There are many copies of that gene product, and that finds its way to the cell surface. It's a slightly artificial system, but you have to do this to be able to monitor changes in ion flux. An ion channel can be gated and controlled, and hopefully you've picked this up in your pharmacology, or otherwise. The way that one way, and this is only one way in which ions are detected, and this is the easiest way to think about it. If an ion concentration changes due to an ion moving from the outside of a cell to the inside of a cell via an ion channel, if it's therapeutically desirable to block that channel with a ligand or a new drug, we can do that, and we can monitor the presence of ion, or the increase of ion concentration within a cell line that's not blocked and a cell line that is blocked by things like phosphorescence or fluorescence, particularly fluorescence, and we can have a metal ion react to a reagent that's internalized within the cell that will show a fluorescence difference in response to a change of ion concentration. Enzymes, this is an easy one to understand. Enzymes enzyme assays can employ surrogate substrates, and these substrates may be engineered to have, these chemicals may be engineered to release fluorescent products. Therefore, your substrate may be non-fluorescent. It gets changed by the enzyme and into products, one of which is fluorescent. That can be detected. And if you inhibit the enzyme, again, a change in fluorescence can be an indication of enzyme inhibition, if the enzyme inhibition is the thing that's required for a therapeutic outcome. This is slightly trickier. There are nuclear hormone receptors such as the estrogen receptor, androgen receptor, mineralocorticoid receptors, glucocorticoid receptors, and so on, that are becoming, or have been, or are becoming, like the glucocorticoid and the mineralocorticoid receptors, becoming very interesting drug targets. And what sort of assay that's used there is often a cellular based assay, in which genetic information includes the transcription of a reporter product that gets a fluorescent outcome. This is activated by the nuclear hormone receptor in presence of a ligand, and if you want agonism, in other words, you want a drug that agonizes or turns on this gene process, you would get fluorescence or increased fluorescence if it's a powerful agonist. If you want to block that for antagonism, then you would look at a control versus presence of an antagonist, and fluorescence would be decreased. So I think that, even though it's a slightly complicated example, I think it's easy to see how that might work. GPCRs are a little trickier. GPCRs, and these are major pharmacological targets. G-protein coupled receptors have many assays, one of which is, although this is slightly being outdated now, but the other assays are slightly more complicated to explain, so I won't go there. GPCRs can be, or ligands or new drugs for GPCRs can be discovered by fluorescent displacement assays. So for instance, if there is a known drug that's or drug analog that's fluorescent, that is known to bind to the GPCR of interest, and you're trying to discover new entities, if we challenge a concentration outside of a cellular system of a ligand that is known to bind to GPCRs, if our new drug comes in and pushes this ligand off, we can get fluorescent techniques that show fluorescent changes in a system that will report the drug potential of our red ligand here. Compound sublibraries. So what we have here is a way of screening drugs against various 96-well plate formats that include ion channels, separately enzymes, separately nuclear hormones, separately GPCRs, for instance, and we would screen chemicals against all those potential targets and evaluate their drug potential, in the attempt to discover new drug entities. Compound sublibraries are used to take a series of compounds that you might be interested in. Those compound sublibraries may be of interest specifically for ion channels, or specifically for GPCRs, specifically for nuclear hormone receptors or enzymes. And those compound sublibraries can be used to target the screening more effectively. And maybe different assays will exist when you start honing in or focusing in on a set of compounds that are very much of interest to you, you may use different assays to improve the quality of the information about how they work at ion channels or how they work at enzymes. Rather than just indicating activity, you might be asking what sort of activity is it at the ion channel, or what sort of activity is it at the enzyme. So for instance, at an enzyme that's of interest to you, if you've now got a series of compounds that show activity against that enzyme, you may be now more interested about what type of activity. Is it competitive inhibition? Is it non-competitive inhibition? Is it covalent or non-covalent, and so on. So compound sublibraries can also be useful to try and aid the simplicity of the problem. Both the previous slide and this slide, if you think about the process, screening multiple compounds against multiple targets and then subsets of those compounds against different the same targets but with different assays, the difficulty of tracking the information in high throughput screens is a real problem. And this is achieved by barcoding libraries and really knowing what's been tested against what in very strict monitoring and information tracking systems. You have to know, and you have to be 100% sure what compound has been tested against what assay, and what information you've got must be linked absolutely rigorously to that test for it to be useful. Chemogenomic approaches, we mentioned earlier, that is throwing chemicals at particular targets to elucidate their structure and their function. That can be done as well. So when you've got a protein that you suspect may be, say for instance, a kinase, you can use a compound sublibrary of kinase-like inhibitors to try and probe if that protein is a suspected kinase, what type of kinase is it, what class does it fall into. And these challenges of an enzyme and its action, let's say for instance an enzyme and its action, these challenges can inform the proteomics. In other words, the function of that protein and what it might be doing in the body because of the type of chemicals it responds to. These are known as screening chemogenomic approaches. So what are these drug targets like? GPCRs are membrane bound, and that's worth knowing. Nuclear hormone receptors are soluble proteins. Enzymes are mixed, they can be membrane bound or soluble. And ion channels by their nature are membrane bound. The consequences of this is that GPCRs, for instance, or membrane bound systems are difficult to get 3D information on. If you've got the purified protein, it may be that protein requires to be in the membrane for its structure and function, and because they are stabilized by the membrane and the biological system has developed for that protein in a membrane, it is often difficult to get the protein out of the membrane and crystallize it for structural studies. So computer modelling may be useful in that instance, and we may well see an example later on where computer modelling is used. Soluble proteins are highly desirable due to their relative ease by which crystals may be obtained, and these processes to obtain crystals are now carried out by automated high throughput techniques to find the right conditions to crystallize proteins, which used to be much more of an art. It's now much more scientific and rigorous. systematically changing conditions under high throughput methodology to achieve crystals. NMR can also throw some light on structural characteristics of our protein targets by looking in membranes, in the solid phase, in crystals, or in solution. In terms of ease of interpretation of information, solution phase NMR is better. Membrane-bound NMR is can be achieved, although it's tricky technique. And then solid phase NMR, again, it has its own technical problems, but solution phase NMR is probably the most desirable. We can take genetic information, we can take a DNA sequence separately, we can introduce that DNA into some sort of microbial vector, and that allows us to clone the gene up, because microbes propagate clonally and reproducibly quickly. That allows us to express the protein, and expression of the protein, this process of DNA cloning and expression is an art stroke science in its own right, and requires scientists to get this effective. Expression of the protein allows us to then purify a protein. We can take that purification that occurs and hopefully take our protein, and maybe if our protein is one that is amenable to crystallization, we can crystallize the protein by high throughput techniques. Once we've got that structure, once we've got that protein crystallized, we can store the proteins by cryopreservation, so they can be transported from site to site if necessary, or stored for further studies, and this is an important process. And these crystals that can be preserved are then exposed to data collection techniques which may be on site, such as X-ray crystallography, or it may be at a remote site, which is why you might want to preserve the protein. Proteins, if they're not cryopreserved, can go off, same as any biological material. They can oxidize, they can decompose, and so on. So data collection can lead to data analysis, and data analysis of the structural characteristics of the protein can lead to a final model of the protein, which then may be used in drug discovery procedures. Okay, once we've got a structure that we want to base a drug design effort on, we can enter into what's called structure-based design, because we have that structure to base design of new chemical entities, new drug entities on that. So the crystal structure of the protein gives us a chance to start a drug development program in a design fashion. We locate the protein active site in our target, and there are various ways of doing that. Either we know the residues that are involved in the active site because of its similarity to other proteins of known structure and function, or we can identify binding sites on the crystal structure by software analysis techniques. We can actually take crystal structures and take crystal structures and actually bind new drugs to those crystal structures and derive a crystallized complex. And we can do this by taking the original crystal structures and soaking the crystals with drug solutions, and hope that our drug binds to the protein crystal and is located in the active site of interest. You would probably want to look for compounds that will bind to the binding site first by computer-aided techniques before you enter into this slightly more lengthy procedure. You may have an idea of lead compounds from some chemo chemogenomics type approaches. In other words, you can take your crystal. Let's say, for instance, your crystal is an enzyme and you have an appropriate assay to monitor enzyme activity, and you start throwing compounds at that protein, at that enzyme, and you can look for inhibition. You can come up with lead compounds that give you some indication of activity that you might find useful for developing towards a drug. If those compounds are such interest that you want to crystallize a complex, then you can pursue that experimentation. Or, and this is often the way nowadays, is that if you think you've identified your protein active site and you're confident that you know that's where drugs might bind, and this is a high percentage of the time, protein active site models in the computer can be generated, and molecular modelling of these systems allow you to do virtual library screening. So you can take virtual screening and dock compounds into that site to see if you can find where they would dock, and they may indicate preferred positions within an active site. Once you identify compounds that might be useful, you can actually go into the real experimentation cycle and crystallize complexes to take a lead compound forward. Generally in proteomics, looking at proteins and their structure and function, proteomics by definition is the not only it includes the identification, i.e. sequence, structure, quantification of proteins, but also the determination of where they're localized in an organ or in a cell, how they're modified, because you can get post-translational modification of proteins, and you should be aware of this, and you've probably been introduced to this at an earlier stage. Their interactions with other proteins, their activities on their own and with other proteins, and ultimately some idea of the function of those proteins, which you need to understand if you want to validate that protein as a particular drug target. And there's a little bit more of a link to those things. Proteomics has other uses, and this slide is actually quite old now, and thank you to the Scientific American magazine to provide the original image, which I've edited somewhat. But proteomics is also used by scientists to investigate proteins that may be interesting drug targets by comparing normal samples with maybe a pathological situation. So here, in this blue slide, we have an example of a male subject with a normal skin sample and somebody that's suspected to have a melanoma. You can take those tissue samples and you can perform things like gel electrophoresis, a technique that you may have come across before, and actually look to compare protein samples within those two skin samples and see where positions of proteins vary, or in fact are they absent, but more likely that they've got a shifted position. From that information you can take a protein and isolate it from a gel directly. You can take that protein and do analytical techniques on it, such as high resolution mass spectrometry. You might be able to get nuclear magnetic resonance out of it. You can also take that protein if you have sufficient amounts of it. The chances are, if that protein becomes of interest, you would sequence it and you would probably clone the protein and now overproduce it within maybe a cellular line and isolate the pure protein from there, and then you can take it into the sequence that we mentioned before, which is to try and crystallize it, determine the structure, crystallize it, perform X-ray diffraction analysis on it, and then get a structural model that will allow you to design drugs against an abnormal target. Proteomics are also used to investigate drug side effects. So you can take bloods from a person, similar subjects, from a person not taking the drug and someone taking the drug. Obviously you have to be worried about their medical backgrounds and the ethics of doing all of this, but certainly their medical backgrounds. They can't be on other medications, they can't have known pathologies, so they both have to be reasonably normal. You allow one of the subjects to take an investigational drug and you test blood serum, for instance, to see the presence of proteins in the serum, and you can compare the two samples to give you an idea of what drugs side effects are, and within a limited subject number, you might decide that the potential side effects are too large to take into a larger population. Obviously, all of this approach requires consent of the people taking the investigational drug. Drug development in the information era. So we've got all these approaches. We've got sequences of proteins to bear in mind. We've got the genes they come from. We've got the RNAs that are produced from the gene, and we've got lots and lots of information to try and track. So bioinformatics is incredibly important. Bioinformatics is the creation and development of advanced information and computational technologies for problems in biology, most commonly molecular biology, but also increasingly certainly in other areas of biology also. And bioinformatics databases can be accessed via PubMed. So PubMed you may have used for your literature searching on topics that you may have encountered to date. But what you may not know is you can take the PubMed tag, which is to the left of the search window, and you have access to lots and lots of databases that contain information about proteins, nucleotides, their structure, the genomes they come from, their conservation, conserved domains within the proteins, epigenomic status, what happens at epigenetic level control of those genes. There's lots and lots of information nowadays that's accessible to anyone. As such, bioinformatics deals with methods of storing the information in a friendly and accessible way, retrieving and analysing that data in the most friendly and obvious way, again because it has to have ease of use. The data is, like I mentioned before, DNA, RNA or protein levels, and the structures, functions, pathways and genetic interactions of all of these components are useful to know, particularly if you have an orphan protein that you're trying to attach a function to and a status within the human. Chemoinformatics deals with chemical information, and it's the application of computer technology to chemistry in all of its manifestations, right through from physical chemical information about a molecule through to its 2D structure, its availability commercially as a chemical, its status as a drug or an experimental, its 3D shape, and so on. Much of the current use of chemoinformatics techniques is in the drug industry, and there's a link to that. Bioinformatics' main role in the drug design process is primarily through ready access to genomic, transcriptomic, proteomic and metabolomic or metabonomic data. That's data of the metabolism within a particular organism. Genomic data is DNA sequence, its protein coding and gene function, structure and control. Transcriptome is RNA structure and function, and whether the RNA is transcribed and how it's controlled and things like that. The proteome has one-dimensional, i.e. sequence data, two-dimensional, whether it's likely to form 2D, secondary structure elements such as helices, beta sheets, random turns and so on. What its 3D structure is likely to be, either determined by X-ray experimental or neutron diffraction or other structural techniques, or by structural prediction, and indeed some information sometimes about its 4D or its temporal expression, sequence of expression in the order of events within the cellular system that you're interested in. The metabolome is metabolic data, and the ultimate goal of metabolomics or metabonomics is to elucidate metabolic pathways on a cell type basis, because the metabolism that occurs within different cell types differs. So normal metabolism of a brain cell is different to metabolism in the liver and so on. And this sort of information is very valuable. Note, all of these omics or these databases that are available can be accessing human information, but they also can be comparative. You can get organism variation, as we mentioned before, common organisms to perform testing on, things like the mouse, Drosophila, the fruit fly, Candarabdidus elegans, C. elegans, the little worm, nematode, zebrafish, and so on, have all been used for experimentation and may throw light on the place of a protein within a pathway of therapeutic interest.